So today I want to finish up talking about carbohydrate structure, talk about disaccharides, polysaccharides, and some modified sugars, and then I will turn our attention to glycolysis. And so it's in glycolysis that we'll start talking about the phenomenon of metabolism, and metabolism being the reactions that occur inside living cells. And so it's metabolism that a lot of people think about when they think about biochemistry. But in order to understand metabolism, we have to understand how enzymes work, and we have to understand how the cell works, and we have to understand something about structure. So we're just about to that point now. We're going to jump right into metabolism, and most of the rest of the term, we're going to be talking about metabolic pathways, and so that'll be important for us to consider. What I want to talk about first, though, is, as I said, talking a little bit about structures. So last time I started talking about reducing sugars, and I'll remind you that a reducing sugar is a sugar that is readily oxidized. So for something to be reduced, something else has to be oxidized. So a reducing sugar is a sugar that's readily oxidized, and the oxidation of that sugar causes something else to be reduced. If I take a sugar and I reduce it, instead of oxidizing it, I create something called a reduced sugar, and these are commonly called sugar alcohols, and specific ones are called alditols. So from the name you can get what it is. That's an aldose that has been reduced to an alcohol. So if I have a reduced sugar might look something like this. There's sorbose, okay? And if we look at sorbose, what do we have? Right, left. All right, we have right, left, right with a ketone. What would that be? Nobody's memorized their structures yet. That's fine. This would actually be fructose. So that's fine. It's not a pop quiz. It's not a pop quiz. All right. So it is a ketose, okay, obviously here. That would actually be fructose. And so if I reduce that, what happens is I'm reducing a ketone to an alcohol. So notice that I no longer have a double bond oxygen anywhere in here. I've reduced the double bond oxygen to an alcohol. If I have xylitol, I can make it from cellulose, right? Well, these guys, in fact, this guy and this guy, both are used for artificial sweetening purposes. Okay, artificial sweeteners. These guys can be metabolized, unlike some of the other artificial sweeteners that are out there. They don't get metabolized very efficiently, which is one of the reasons that they're used as artificial sweeteners, because they don't generate a lot of calories, at least not as much as the native sugar generates. This one in particular, xylitol, is used as an artificial sweetener. You commonly see it in things like breath mints and various fresh things in your mouth, when you see them advertised as fresh things in your mouth. The reason that xylitol is used is that when it dissolves, it actually absorbs energy. And so when that happens, absorbing energy to be dissolved means that it tastes cool to you. So when you get a cool, refreshing taste from something, it's happening commonly because it's got xylitol in it. All right. Now, sugars can be modified in a variety of ways. I talked yesterday briefly about how we can make glycosides. And glycosides are made by modifying the anomeric hydroxyl. So here's the anomeric hydroxyl. We see here that a different portion of the molecule is modified. In this case, carbon number 6 gains a phosphate. And we're going to see that when we talk about glycolysis, this is actually the very first reaction of glycolysis, putting a phosphate onto glucose. That makes a phosphorylated sugar. Okay? And this is not a glycoside because the anomeric hydroxyl is unaltered. Okay? So that means this guy could go back and be a linear and then flip alpha, beta, etc., because it's not blocked here. If this were somehow altered, that could not happen. Yes, Mariah? So a glycoside, her question was, what happens in an anomeric hydroxyl and a glycoside? A glycoside is defined by anything that modifies the anomeric hydroxyl. So the example I gave yesterday was I put a methyl group on here. Well, that's altered the anomeric hydroxyl, and if I do that, it's now called a glycoside. Okay? Laetrile was an example of something I showed that had an altered anomeric hydroxyl. Okay? This one's not a glycoside because the anomeric hydroxyl is not altered. Okay. And there's, just to refresh your memory, there's Laetrile. So there is a sugar. There's the anomeric hydroxyl, but it no longer is a hydroxyl. It's had something put onto it. So that becomes a glycoside. And there's a lot of examples, zillions of examples of glycosides that are out there. We'll talk about many of the polysaccharides and disaccharides are also glycosides. So we can think of glycosides as not necessarily being things that are poisonous or bad for you, but that they're just simply a structure that is commonly present. Okay, ascorbic acid. Ascorbic acid is vitamin C, and you don't need to know the structure or anything like that, but you can see it's a fairly simple molecule, and you can imagine that simple molecule could be, in fact, derived from a sugar. It turns out that ascorbic acid can be derived from glucose by some organisms. We can't do that, but some organisms can make ascorbic acid or vitamin C from glucose. And if we count the carbons here, I always encourage you to count the carbons. I've got 1, 2, 3, 4, 5, 6 carbons present, right? And so I know that it's not crazy to say that it might be modified from glucose, which also contains 6 carbons. Yes, Clara? Why can we not make it from glucose? We don't have the enzyme necessary to do it. So whenever we talk about being unable to make a molecule, it's because we lack an enzyme necessary to do that. Us and rats are 2 mammals that can't make glucose. Most mammals can actually make glucose. Yeah. Make glucose. Make ascorbic acid, I'm sorry, from glucose. But we can make glucose, sorry. It's Wednesday is the Friday of this week. It's the first Friday of this week for us, so my mind is in Friday mode at the moment, so I apologize. Okay, disaccharides. By the way, a saccharide. What's a saccharide? Well, a saccharide is the same thing as a carbohydrate. Okay, saccharide derives its name from the fact that it means sweet tasting. That's what the word saccharide literally means, is sweet tasting. So we have a monosaccharide. Glucose is a monosaccharide. Fructose is a monosaccharide. Okay, those monosaccharides have 1 unit. When we look at disaccharides, not surprisingly, they have 2 units associated with them. Okay, 2 units. So we can see that these 2 units can be combined in a variety of ways, usually involving a glycoside. Usually. So as soon as I say glycoside, I want you thinking anomeric hydroxyl. And is the anomeric hydroxyl altered? And look, there's an anomeric, what was a hydroxyl. It has been altered. Therefore, this guy is a glycoside. We call this a glycosidic alpha-1,4 bond because we look here and say there's carbon number 1. And carbon number 1 has the hydroxyl pointing down. So that's an alpha position. 1 linked to carbon number 4 of this one. There's carbon number 1, 2, 3, 4. Make sense? It's an alpha-1,4 bond. What is this molecule? Down, down, up, up. That's glucose, right? What's this molecule? Down, down, up, up. That's also glucose. This is a disaccharide of glucose. And the name of this one actually is maltose. You don't need to know that, but that's what that is. Here's something that has an alpha-1,6 glycosidic bond, right? Down, down, up, I made it easy. Down, down, up. It's 2 glucoses here. Now, notice how we had to draw this to make it look right. So people draw these sort of shorthand structures indicating it's going down, right? It doesn't literally bend like that. It literally is positioned more like this. But to fit it onto a screen, that's the way people draw that bond, is so that it's pointed down like that. But it's an alpha-1,6 because, again, alpha position, there's 1, and there's carbon number 6 up over there. Yes? The fact that the anomeric carbon is going down, that's what an alpha is, whereas a beta is when the position is going up. Okay? So I'm drawing this down to indicate it is an alpha bond. Okay? And this one clearly is the same thing as well. Okay? Okay. Let's see. Oh, sorry. Before I do that, let's go to sucrose. So This highly artistic drawing was made by me to illustrate the structure of sucrose. Okay? Now, I haven't shown you the structure of fructose, so I need to introduce that to you first. Fructose is a hexose, has 6 carbons. However, it's a hexose that exists in a furanose form. So if you remember what I said yesterday, a furanose is something that contains a 5-membered ring. So if we look at this, well, this guy has a 6-membered ring. This guy has a 5-membered ring. You could probably infer from what I'm saying that the bottom one, in fact, is fructose. What we're looking at on the screen is the structure of sucrose. Sucrose contains a glucose, and it contains a fructose. Okay? The fructose, notice the structure of the fructose. It's a 5-membered ring, oxygen here. Carbon number 2 is right here. Now notice the oxygen is bound to carbon number 2, not to carbon number 1. Does anybody know why? It's a ketose. And the ketose had the double bond oxygen on carbon number 2. Glucose had the double bond oxygen on carbon number 1. So there's the bond to carbon number 1 that was the double bond oxygen before. Here's the bond to carbon number 2 that was the ketone before. Now, if we look at this, what do we see? There's carbon number 1. Don't forget carbon number 1. Carbon number 2. Carbon number 3, 4, 5, 6. Fructose has 6 carbons. Don't forget that. Yes, sir.
Mechanistic question.
Yes.
Does cyclization occur before or after the glycosidic bond?
Very good question. Does cyclization occur before or after the glycosidic bond occurs? It occurs before. It occurs before. Yes?
Like, so sucrose is fructose
And glucose. Now, let's think about that bond that Mariah just asked about, okay? If I said to you, how would you describe this? I would hope that you would say it's a glycoside, right? Anomeric hydroxyl is messed up. But this guy turns out to be a diglycoside. This anomeric hydroxyl is messed with. But this was a hydroxyl in fructose. So now I say, okay, so the hydroxyl, the anomeric hydroxyl of both carbons was messed with. This is a diglycoside. It's unusual. Okay? If I had the structure of fructose, I would have all of this, and this would be an OH. Now, fructose is one of the molecules you need to know the structure of. You need to know the ring structure and the linear structure. I showed you the linear structure yesterday. You need the ring structure and the linear structure of sucrose. Is this alpha or beta? I hear one vote for alpha. No, for the fructose. Okay, so it looks tricky, doesn't it? Because this guy's pointing down. However, it is the hydroxyl that determines the alpha beta. So the hydroxyl is up. This is the beta form of fructose. Right? So it's always the hydroxyl that determines whether it's alpha or beta. So when the hydroxyl is up, it's the beta form. So we have glucose in the alpha form. We have fructose in the beta form. Okay? And we put those together and we have sucrose. What does sucrose do? What does sucrose do? Well, sucrose converts this guy, which is an aldose, into something that's no longer an aldose. And it can't flip back to an aldose because it's a disaccharide. It's a diglycoside. Why is that important? What did I say about how easily aldoses were oxidized? It happens relatively easily. This is actually protecting this thing from becoming oxidized. You might wonder why we have high fructose corn syrup. One of the reasons that companies like high fructose corn syrup is fructose is less readily oxidized than is glucose. Okay? That's one reason. Yes?
How are they diglycosides if they're attached by 3 proteins first carbon and fructose is second carbon?
Okay. How is this a diglycoside? Okay. If I had glucose, this would be an OH, right? If I had fructose, this would be an OH.
But is that the anomeric
It is. It is. So the anomeric hydroxyl of each one has been altered.
I thought the anomeric hydroxyl comes off of the first carbon.
No, the anomeric hydroxyl comes off of the carbon next to the oxygen. Yeah, yeah. So there, you can see there's the anomeric. It's the one that forms when the ring structure forms. And this forms when that ring structure forms. Good question. Okay. Let's take a look at this. Here are some disaccharides. Okay. Lactose, that's known as milk sugar. You don't need to know the structure of it, but lactose, you should know, contains glucose and the sugar galactose. Glucose and galactose. Okay? Maltose, I showed you earlier. It contains 2 glucoses. And they're linked alpha-1,4. Again, you don't need to know that, but I'm just showing you that structure. But you should know that maltose contains 2 glucoses. You should also know that sucrose contains glucose and fructose. Yes, ma'am?
Does the glycoside in maltose prevent oxidation?
Okay, does the glycoside in maltose prevent oxidation? What do you guys think? Nobody has an opinion? It prevents oxidation here, but notice what do we have out here? This is still has the potential to go back and become linear and become an aldehyde. So this one will not be protected on this side. So one side it is. So sucrose is unusual. Both of them get protected by that diglycosidic bond. Most disaccharides and most polysaccharides have at least one anomeric hydroxyl unaltered. Yeah, important point. Yes, sir? Yes. Good. Good question. You're ahead of me, Nick. So one is glucose, fructose, linear and straight chain, sucrose. Sucrose only exists in the, I said linear and straight chain, linear and circular, okay, and sucrose only exists in the circular form. Right? Okay. Now, the other one, and I just realized I don't have it, a figure showing here. I'll post one for you. You should also know the structure of ribose. Ribose is the easiest of all of them to remember. But since I don't have a structure, I'm not going to try to describe it to you. You'll see when I show you. You should know the linear and the linear and the circular forms of ribose. Ribose is important because, of course, it's the sugar that's found in ribonucleic acids. ATP, GTP, CTP, UTP all contain ribose, and so it's important to see that, to know that structure of ribose, and I'll post that for you. And those are the only ones you're responsible for the structure of. Okay, sure. Okay, here's an artificial sweetener. It's called Sucralose, and the company that makes Sucralose says it tastes like sugar because it's natural like sugar. What does it mean to say it's natural like sugar? Well, the best I can figure is because it has a form of fructose linked to a form of glucose, and therefore that somehow makes it natural. It has some very unnatural things in it. That chlorine replacing a hydroxyl is a significant change. That chlorine replacing that hydroxyl is a significant change, and that chlorine over there is a significant change. Why did they make those changes? They make those changes so that the enzymes that break down sucrose can't act on it. If it can't act on it, then you can't burn it for energy. And if you can't burn it for energy, there are no calories. And it tastes sweet, so everybody's happy, right? Well, maybe, maybe not. As far as I know, there aren't any studies showing problems associated with sucralose. So what I'm going to say is only my own speculation. But I will point out something that we don't see in biology almost anywhere, and that is a carbon chloride bond. We just don't see it. You're not going to see it in any molecule I'm going to show you. Okay? And one of the few places we actually see such a bond is in dioxin. Dioxin is considered a pretty nasty compound. Now, I'm not saying this is dioxin, because if it were, we would have a lot of dead Americans right now, or dead people around the world. However, when I see that bond, it causes me a bit of worry. Okay? It causes me a bit of worry. All right? This compound has an interesting story associated with it, which I'll tell you. This compound was developed in England. And in England, the company, the group that was making this was trying to make an insecticide. And the idea that I'm telling you here about the carbon chloride bond made a lot of sense, right? Dioxin has a carbon chloride bond. Maybe we can make something that'll kill insects. Okay? So they spent a fair amount of time in investigating how to make this compound. And when they made the compound, the graduate student in the laboratory calls the professor and says, hey, I've made the compound. They didn't call it sucralose at that time, but I've made it. And the professor got the message, and he calls back, and he can't get the student. So he leaves a message saying, OK, test it. Right? So what do you suppose the student would do? He would go out, he would get some insects, and he would try to see if it works. But the student got the message and thought he said, taste it. And he did. The importance of communication is high here, folks. Imagine you've been spending all your time making an insecticide, and your professor says, taste it. That's an act of faith, isn't it? And he calls the professor back and it leaves a message and says, it tastes sweet. And the professor says, huh? You idiot! I said, test it, not taste it. Those British accents just get you, right? It's like, I don't know what it was, but can you imagine that? Right, so anyway, that's the story of one of the stories of sucralose. Test it. Okay, polysaccharides. Let's spend a little bit of time talking about them. I'll go through those fairly quickly. Polysaccharides, as their name suggests, contain multiple sugar units. They will almost always have multiple glycosidic bonds. This is the structure of a polysaccharide known as amylose. Amylose is a very common polysaccharide in plants. It's used as a storage form of glucose. By the way, one of the things you're going to hear me say over the next few days is glucose is a poison. Glucose is a poison. Your body and every cell on Earth treats it as if it's a poison, but it's a poison that they need to have to make energy, meaning that they will tend to keep glucose in low concentrations. Glucose in high concentration causes a cell to swell, take on water, and explode. Keeping glucose concentrations low is therefore important. Okay? One of the ways to keep glucose concentrations low is to keep it in a polymeric form. This is not poisonous. You know glucose is poisonous because I'm sure all of you know people with diabetes. And what does diabetes arise from? High blood glucose. And high blood glucose causes damage to organs for the reasons I just told you. The reason your body has insulin is to manage blood glucose so that it doesn't get too high. Now, glucose is a double-edged sword because you can't let it get too low either because you need to have a certain amount for energy, right? But glucose is a poison. I want you to remember that. So the way of dealing with this poison in a plant is to put it into a polymeric form. Look at the bonds. Alpha 1,4. Alpha 1,4. Amylose is a very simple structure. It's simply a polymer of glucose units in the alpha 1,4 configuration. You can see this can be 300 to 600 long. Okay? Long polymers. Okay? By the way, we can digest this very easily. We have enzymes in our saliva known as alpha amylase that will start breaking this down as soon as you put it in your mouth. As soon as you put it in your mouth, your enzymes in your mouth will start breaking that down. Clara. Okay, question, is this a glycoside? What do you think? Yes. Will any part of this be oxidized? This guy could be because it's an unaltered anomeric, and that's going to make an aldose. Yes, could be. Well, okay, her question was, will anything above a monosaccharide be a glycoside? They usually are, but they don't have to be. What if I had a disaccharide made by joining the hydroxyl of carbon 3 to carbon 4 of another sugar? You would not have an anomeric involved, So it doesn't have to be. But it would be unusual to see a sugar like that. Okay? Okay. So how many is a polysaccharide, by the way? Usually we start talking about polysaccharides once we get up above about 4 or 5 sugar units. We just call them a polysaccharide. There's amylose. Here is the primary energy, primary glucose storage form that we use in our body. It's called glycogen. Okay? Glycogen is also a polymer of glucose, but it's got an added wrinkle. Okay? Notice if we leave the top part out, we just look at the bottom, what do we see? Alpha-1,4 linkage, alpha-1,4 linkage, alpha-1,4 linkage. We have a lot of these. We keep glycogen in our liver, and we keep glycogen in our muscles. We'll see later why that's so important in our muscles. Okay? But glycogen has a wrinkle. The wrinkle is glycogen has something we call branches. Here's a branch. Notice this is an alpha-1,6 branch. And we go down here, and here's another alpha-1,6 branch. That's what this is designating, branch points. So glycogen is a polymer of glucose in alpha-1,4 linkage with alpha-1,6 branches. And those branches in our body occur about every 10 glucoses, about every 10. Notice that this continues on. This branch will continue now in alpha-1,4, 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 alpha-1,4. This will continue alpha-1,4, alpha-1,4. So each one of these can go off and make additional alpha-1,4s. Yes? This is glycogen.
Why is it linkage like this if we keep glucose in linkage to keep our glucose levels down?
Okay.
Does that make sense?
Not quite sure I understand, but let me just say a word. We are keeping our glucose levels down because it's in a polymeric form, so we don't have free glucose. Okay? And we want to be able to break this down readily because we use this not just to hide glucose, but there's times we need to use glucose. So our muscles, for example, if I take off running, I will very quickly exhaust my ATP. And how do I get more ATP? By burning glucose. And if I have a storage form of glucose, I break this down, I get glucose that I can then burn for ATP. Okay? The reason we have this in our liver is our liver plays a very important role in modulating blood glucose levels. Not muscle levels, but blood glucose levels. Our brain needs glucose. Our eyes need glucose. They can't make it. They are totally dependent upon our liver to provide the right amount of glucose for them to function. Too low, we faint or die. Too high, we have diabetes. Okay? So the liver has to play that very important role in modulating that. But I want to emphasize the structure here. The structure, we have a basic component and we have branches that come off of there. If we think of this as several hundred or several thousand units long, we could have branches that go, we could have hundreds of branches, and each of those branches could have branches. Right? So we start thinking of a very complicated structure. The main thing I want you to take away is that complicated structure, each one of them has an end. This guy eventually is going to come to an end. This guy is going to come to an end. This guy is going to come to an end. All right? I could have thousands or hundreds of thousands of ends. Why is that important? Not to be oxidized, that's a good thought, but the only oxidation that will happen is at this end. There's only one end that can be oxidized, and that's where the anomeric carbon was. These guys are going to end in free carbon number 4s. Okay? The reason that the ends are important is it turns out that glycogen is broken down starting from the ends. And so we use glycogen as a quick source of glucose, and the enzymes start at an end and move inwards. Well, if we have a lot of ends, we can release a lot more glucose really, really quickly. This is why we have glycogen, and plants have amylose and a related one called amylopectin that I'll describe in a minute. Those molecules have either, in the case of amylose, one end, Amylopectin is like glycogen, except for its branches don't occur nearly as frequently. They'll have branches about every 50 or so. Plants have amylose and amylopectin. Why do they have much less branched storage forms of glucose? Plants don't have muscles. Plants don't run away from things. Plants don't need immediate energy. So at night, when there's no photosynthesis and no production of glucose going on, plants say, okay, I'll get a little glucose to keep me going. That doesn't change drastically. For us, if a grizzly bear comes out of the woods and starts chasing us, I want my glucose now. Right? I want it now. I don't want it to kick in, 3 hours after midnight. I want it now. So we have immediate needs for glucose, and so we have more branching as a result of that. Before I talk about starch. So starch. Starch is a common polysaccharide in plants, and it's actually not a compound. It's a mixture. It's a mixture of amylose and amylopectin. So amylopectin is just like glycogen. It's got branches, but it only has branches about every 50 glucoses or so. This is good because it shows you this branch. Here's the branch. There's an alpha-1,6, and then notice everything else here is alpha-1,4, alpha-1,4, alpha-1,4. So only the branch point is alpha-1,6. Only the branch point. Cellulose. Cellulose is also a polymer of glucose. Except for instead of having alpha-1,4 bonds, it has beta-1,4 bonds. Cellulose is important for the structure of the plant cell wall. Plants have elaborate cell walls. We don't have elaborate cell walls. Okay? The beta-1,4 bond cannot be broken by any enzyme in our body. Cannot be broken by any enzyme in our body. Therefore, plants for us are roughage because there's undigested parts of them that pass through. And they help with things passing through. Ruminants, on the other hand, such as cows, can break down cellulose because they have an enzyme in a bacterium in their rumen called cellulase. Cellulase. Cellulase breaks beta-1,4 bonds of glucose. Of cellulose. It breaks beta-1,4 bonds of cellulose. Consequently, ruminants can get energy from eating grass, eating any plant tissue. Okay? We can't get energy from breaking down cellulose like ruminants can. Yes, ma'am? Is this why some people eating vegetables can get bloating? That can be one cause, yes? Mm-hmm. Yep. Depends how much you eat. Yes. Okay, I just realized as I was saying that there was one other enzyme I started to talk about before and I didn't finish, and that was lactose intolerance. So let me come back up and say a word about that. When I talk about lactose, people frequently ask me about, well, what's lactose intolerance? Okay, because we hear about that. Lactose intolerance means that many people, as they get older, produce less of the enzyme that breaks down lactose. The enzyme that breaks down lactose is known as lactase, and these are enzymes that are easy to remember. ASE always indicates an enzyme. OSE always indicates a sugar. Okay? Lactase breaks down lactose into glucose and galactose. Okay? Lactose intolerance arises because if you don't have that enzyme, lactose, which gets normally broken down by lactase early in the digestive process, makes it further into your digestive system, where there are little tiny bacteria that love lactose and produce prodigious amounts of gas. It'd be very uncomfortable. It'd be very embarrassing, right? Consequently, that's why that is a problem. If you want evidence, anybody ever asks you for evidence of evolution, here's a really good thing to talk about evolution of human beings. Okay? We know on the evolutionary tree when human beings started making lactase as adults. Early human beings didn't do that. Why? because early human beings only drank milk when they were infants. Okay? We know from the evolutionary record when humans had a mutation that they started making lactase as adults. And it happened exactly when farming came about. Domestication of animals. And having milk available at times besides when you were an infant. If you didn't have that, you had lactose intolerance, and likely you didn't survive it well. Those that had that mutation went on to give rise to all of us. Now, it's not perfect. Okay? There are people who, and it tends to be ethnically based a little bit. Asians tend to have a little bit more difficulty with lactose intolerance than non-Asians. That mutation is not perfectly expressed. So if you go for a long period without, for example, drinking dairy as an adult, you can actually have your own lactase expression decreased. But that's a really good piece of evidence of evolution in action in human beings right there. Okay, I'm full of stories today. Let's get back here and finish up a couple of things. There's glycogen versus amylopectin. I told you this had a lot more ends. Look at all those ends. All those green guys are ends that can be broken down. Here's a roughly equivalent structure for amylopectin, not nearly so many ends, because this is, again, releasing a lot more glucose very quickly when glycogen is broken down. All right, some other modifications. Here is what we call an amino sugar because it contains an amine. See that NH right there? This is N-acetyl beta-glucosamine. No, I'm not going to ask you to draw that. But I would hope you would know from the N, of course, that it contains an amine. I would hope you'd be able to look at this and tell me, is this a glycoside or not? It's not, because there's the anomeric hydroxyl, and it's unaltered, right? This guy turns out to be a building block of another interesting biopolymer, and that biopolymer is known as chitin. Chitin is abundant. You can see it's a beta 1,4 linkage between N-acetylglucosamine units. Chitin is a polysaccharide or a polyaminosaccharide that is present in the exoskeleton of insects. The exoskeleton of insects. Okay. No, you're not going to draw that structure. Don't worry. Another important polysaccharide is actually found in the cell wall of bacteria. And it's called a peptidoglycan structure. Let's think about what that name means. Peptidoglycan. Peptido referring to peptide. Glycan referring to a polymer of a sugar. Peptidoglycans make a nice sheet, and they're protection against penetration of a bacterial cell. Okay? These guys have polymers of a sugar. You can see the polymers of the sugar going along here. And they have peptides that link them together. That's the smaller units that's in here. Okay? So these peptidoglycans have a sort of I don't want to call them a backbone, but they have a polymer of sugar units joined together by peptides and held together in a sort of a net, like you can see here. Right? I tell you this because when we look at things like penicillin is an antibiotic that works by inhibiting the synthesis of the peptidoglycan layer of bacteria. If bacteria can't make this, they can't divide. They can't make cells, and they will die. Okay. All kinds of terms here. What's a proteoglycan? Okay. All right. Peptidoglycan and proteoglycan, right? They sound like similar things, and they actually are pretty similar things. Here is a structure of a proteoglycan. Proteo referring to protein, glycan again referring to a polymer of sugars. All right? Here is a proteoglycan where we have a protein that has a whole ton of sugars linked to it. Okay? So proteoglycan and a peptidoglycan are basically proteins that are linked to sugar polymers. Proteins linked to sugar polymers. These guys turn out to be important for things like snot. I'll talk about that later. Okay? The mucousy feel, right? That arises from structures like this. Okay? How does, and this particular one, hyaluronic acid, is actually part of synovial fluid. How many people know what synovial fluid is? It's the lubricant of joints. Think about snot and how slimy that is. Synovial fluid works because it's slimy. Okay? That's why your joints actually lubricate well, is because of the sliminess of them. Proteoglycan versus a glycoprotein. I'm not going to hold you responsible for that. Okay? I'll tell you briefly what a glycoprotein is that's different. Okay? But you won't have to tell me the difference between a glycoprotein and a proteoglycan. A glycoprotein is the sort of opposite. We said that the proteoglycan was a protein with a bunch of polymers of sugar attached to it. A glycoprotein flips that. It's a protein to which we attach a short set of sugars. Okay? A short set of sugars. Why do we care about them? Well, I mentioned earlier in the term the importance of identity, cellular identity, and we showed that we had that cell structure, and we had proteins, and we had some short sugars sticking off of them. I said they're important for blood groups. Okay? The presence of specific sugars out there give rise to one type of blood group versus another type of blood group. So here's, for example, a type A blood group antigen. That would have galactose, and this guy shown here. I'm sorry, would have galactose and this guy shown out here. This would have galactose and, where are we at here? Oh, here, fructose. Okay, what am I got? Those look identical, don't they? Oh, well, no, let's see. Why am I seeing that? I think I've drawn two identical ones there. But what the primary differences between blood groups are one difference in the sugar component. And I think I've actually drawn two of the same ones here. But let's say that in an A type, I might have something down here like N-acetylglucosamine instead of fructose. And the reason that's important is that what gives the importance of blood groups is your immune system. Your immune system will attack foreign blood groups. So if I have down here, for example, N-acetylglucosamine instead of the fructose that my body is used to, the body will say invader, and the immune system will attack it. And you die if someone gives you the wrong blood type because of a massive immune response. Yes?
So these glycoproteins are the quote-unquote tags that you mentioned last.
Do they what?
They're the tags that you mentioned?
They are the tags, yes. So glycoproteins provide the sugar tags on the outside of the cell that give the identity of the cell. Okay. That's the last of what I want to say there. And I just want to introduce the topic of glycolysis before I turn you loose. We've been talking about the structure of carbohydrates. And as I said, we're turning our attention to the process of glycolysis and metabolism in general. As we talk about metabolism, we have to start thinking about energy. Energy is critical because glycolysis, for example, gives energy. And glycolysis has energy limitations through which the cell must function. Every chemical reaction has an energy component to it, whether it's requiring energy or whether it's releasing energy. So it's important that we think about energy in the overall scheme of reactions, right? Everybody's seen this equation. We've talked very briefly about Gibbs free energy before. Gibbs free energy, which is this guy here, the change in Gibbs free energy for a reaction, okay, tells us a reaction direction. If the Gibbs free energy for change for a reaction is negative, the reaction goes forward as written. If it's positive, it goes backwards as written. And if it's 0, it's at equilibrium. The Gibbs free energy change for a reaction is equal to a standard Gibbs free energy, which is shown by the delta G0 prime, plus this term. This is the variable of the equation. It varies because products and reactants can vary. This is a constant. We talked about the Henderson-Hasselbalch equation. We said the pH is equal to the pKa plus log of salt over acid, right? The pH varied as the salt over acid varied because the pKa was a constant. This is exactly the same thing. The delta G for a reaction varies as the products over the reactants change because the delta G0 prime is a constant. This is the Gibbs free energy change at standard conditions. In standard conditions only, this is the Gibbs free energy change at any condition. What do standard conditions involve? 1 molar products, 1 molar reactants. If I have 1 over 1 here, what happens to the natural log term? 0. That's why this is the Gibbs free energy change under standard conditions. What happens if I have a reaction, A going to B, and I dump a bunch of A into the mixture? What's going to happen to the reaction? Okay? So the reaction, if I dump a lot of A into there, it's going to be pushed forward, okay, by Le Chatelier's principle, right? So I'm going to make more B. This tells us why. If I put more reactant in, what happens to this ratio? The ratio gets smaller. If the ratio is less than 1, what happens to the log term? And if it's negative, what happens to this? It gets smaller, right? The smaller this gets, the more likely it is to go forwards. So the more A I put into there, the more negative I make this, and the smaller I make this, favoring the forward reaction. On the other hand, if I don't dump in this, but instead my product starts to accumulate, what happens? When the product starts to accumulate, this ratio becomes greater, the log term becomes more positive, the delta G becomes more positive, and the reaction starts going backwards. Right? So we can understand Le Chatelier's principle by understanding the Gibbs free energy. Rather than go through that at this time, I'm going to bring up the song of the day, and then we're going to call it a day. This one I'm going to have somebody else sing for me, because I don't sing it very well. You can sing along, though. Okay, see you guys on Friday.